The decoding of human genome ushered in a new era in biotechnology. Understanding and interpreting terabytes of genome data, predicting the structure and function of tens of thousands of proteins implicit in the genome data, and designing drug molecules for target proteins constitutes some of the grand challenges in science. Supercomputers are harnessed worldwide to tackle such problems. Current-day supercomputers can carry out more than 1 trillion operations per second. To enhance and accelerate Indian contributions to genomics, proteomics, and drug design, a supercomputer has been commissioned at IIT Delhi on July 31, 2002.
The supercomputer at IIT Delhi consists of 35 280R servers housed in these 4 racks. Each server consists of 2 UltraSpark 900 MHz processors and a 2 GB RAM, and through a network card each server communicates with the other. The total storage capacity of the system is over 1 terabyte and the total compute power of the system is over 100 gigaflops.
If a single person can do a job in 100 days, depending upon the job, one can employ 100 people to do the same job in a single day. Modern day supercomputer exploit this strategy.
Clusters of personal computers or workstations can be connected via a switch and made to work in concert, offering a low-cost supercomputing environment.
All the 35 nodes are integrated through a gigabit switch through which they talk to each other.
The supercomputer at IIT Delhi can carry out more than 100 billion operations per second. Life propagates via DNA comprising 4 basic units: adenine, cytosine, guanine, and thymine, connected sequentially and repeated several thousands of times in some yet unknown ways in each cell in an organism. Genome is the DNA content in a cell. Each cell carries this information. Which part of the genome, called gene, gets expressed as a protein in which cell, when and how are some unsolved puzzles. Gene identification, the spatial and temporal issues connected with the expression offer some immediate challenges. The information is out there in the form of genome, we just need to know how to interpret it. Genome data, which is becoming available at an accelerated pace, poses several challenges to data storage with easy access and data mining and eventual product design. The scientific fields connected with this endeavor and related activities are known as bioinformatics and computational biology. Information must lead to knowledge and knowledge to products useful to society. The expectations from bioinformatics in India are very high given the strong mathematical and IT skills. In agriculture sector, genomic information can be utilized to improve volume of the produce, the nutritional content, and to implant disease resistance. In medical sector, genome information can be fed to supercomputers which can then identify drug targets, design effective drug candidates, reduce the cost and time involved in drug development with a long-term goal of developing personalized medicine with minimum side effects. There are 3 major areas of research in bioinformatics: genome analysis, protein structure prediction, and drug design. The IIT Delhi team is developing a chemical model for genome analysis and methodology for protein structure prediction. The protein folding problem is as follows. Given a sequence of amino acids, what is the shape, namely the 3-dimensional structure of the polypeptide chain? Professor G.N. Ramachandran, an Indian scientist, made fundamental contributions to protein structure analysis. Protein folding problem remains unsolved as of date. Structures of proteins can be predicted with ease if similarities exist in databases. An alternative is ab initio method, which typically uses 1 million processors and 1 year compute time to fold a single protein. The team at IIT Delhi is developing methodologies combining the best of bioinformatics and ab initio methods to tackle this problem in a computationally efficient way. Designing a drug for a target is more like making a key for a lock, except that this lock, that's the target site, consists of atoms and it is dynamic. The team at IIT Delhi developed and successfully demonstrated a methodology christened Sanjeevani, whereby the computer can distinguish drugs from non-drugs for a given protein target. We believe that India will make some original contributions in the areas of genome analysis, protein folding and drug design in the years to come, benefiting the entire mankind.